So a couple of questions after I'd already stopped the other recording as it relates to the exam next week, the practical exam. So all along the side there will be microscopes set up, right? And so you'll get up from your seat and you can go and look at each one of the microscopes. But I'm not going to have stations throughout the room. I'm going to actually print up the test. Each person will get a copy, and you'll have an answer sheet that you write your answers in on, right? Remember, you need to spell, right? This is not going to be a Scantron test, right? It's like the standard one that you're used to seeing where it has 50 blanks and you got to fill them in, okay? Make sense? And then I'm going to have a lab bench designated, or part of a lab bench, for you to be able to do the aseptic transfers and focus the microscope. Okay? Does that make sense to you guys? And I think I'll be able to even maybe do a whole lab bench because we could consolidate into 3 where you guys can sit and write. Right? So you don't get to necessarily sit in your seat. Right?
So after each person you go behind them, unfocus the microscope.
Yeah. Yeah, you'd have to start all over again. So for the microscope focusing station, right, we're going to put the stage all the way down when you're done, and the next person has to completely start over. So from scratch. And I realized today for the quiz I didn't do that, so some of you guys probably lucked out. But I didn't take off that many points for focusing. Issues helped you guys out. Note for the next time I do that. Any questions as it relates to the test? Okay. So remember, when you come in, you're going to put on your lab coat for the test, right? And you'll put on your safety glasses when you come to this lab bench to do the aseptic transfers. But you won't need them for the microscope. And of course, proper lab attire. I shouldn't see any skin on your legs. Okay. So today you're going to do 2 stains. You've already made your smears, right? We'll heat fix them and then we'll start staining them as soon as I shut up. So, as I said, it's really important to know what are the 2 methods that we're using for these procedures, because there are more than these methods out there, right? And it really depends, your results really depend on what method you're using. So, for this method, as I said, you shouldn't see this, right? This is on these prepared slide. It's up here, mycobacteria, right? It'll look like, hopefully, this picture. So these are acid-fast bacilli, commonly abbreviated AFB. So they're rod-shaped. They are going to stay stained by the carbolfuchsin. They tend to clump together because they're hydrophobic. They don't like water. They contain, in addition to peptidoglycan, mycolic acid, right? And so this is a very waxy substance. And this is why it's called the acid-fast procedure, because the decolorizer, and this is important, the decolorizer you use for this procedure is not 95% ethanol. You guys will notice that there was another clear bottle on your lab bench today, right? It says acid alcohol. So this is ethanol with 3% hydrochloric acid. You definitely want to wear gloves, right? We wear gloves when we stain anyways, right? But you want to wear gloves. This stuff will burn you, right? It is an acid. And that's why it's called acid fast, right? Because despite using acid, which is a really strong chemical, right? These guys retain the dye. They stay stained with the carbolfuchsin. So fast, in this use of the word, they're meaning it stays, right? When we think of fast, we think of running fast. But fast actually has a different definition that most people don't use, right? In this case, it means it stays. Does that make sense to you guys? Stay fast, right? Like stay fast. So there's its peptidoglycan layer, and then there's the mycolic acid. So lots of carbon associated with it. So anywhere from 20 to 26 carbons, or even 50 to 60 carbons. So these are really long, very carbon-rich molecules. No, you don't need to remember the number. Right? It's just know that it's waxy, right? And that's why they're going to retain the dye, and the reason for that we're going to explain in a moment. So why would we want to use this staining procedure? Because we want to know if an organism is acid fast. Because if it's a bacteria, there's a really good chance it's one of these guys. So Mycobacteria leprae, you would want to know, right, if you had leprosy. Hey, what's up?
I have refrigerator issues. I want to succeed in coming to see if I have space to stow some stuff.
What do you need to stow? Because that's dirty, you know. So you would have to store it in those.
They stay cold.
Yes.
You have a freezer.
That's it.
It's a dirty freezer.
I can semi. I do have it boxed. It's small.
Happy Tuesday, everybody.
Yes. Okay. So, this is a little vignette. I'm not going to test you on leprosy, right? This is FYI, especially you guys going into health fields. So the other one that we really are concerned about that's a mycobacterium is tuberculosis, right? And that's actually the slides that I have for you guys. Of course, they're dead, right? They're stuck on. You're not going to get tuberculosis. But they're all mycobacterium, and they stain acid fast. So some other organisms that will stain acid fast that are mentioned in your book are this Nocardia, and can cause some pretty serious infections. This is a eukaryotic organism that they'll use for staining with the acid-fast stain, although this picture isn't an acid-fast stain. I don't know why Peter picked that one. So sometimes we use acid-fast staining for non-bacteria, but for the most part we're looking for mycobacteria. So when you do this staining procedure today, you can see that it is possible, right? This was done in our lab with our screen, that you can fit, if there's 4 of you at the lab bench, you can fit 4 slides on at the same time. The only problem is even with our wax pencil, sometimes your little labels will get rubbed off. So I suggest writing yourselves out a key who put whose slide where, right? So you can retrieve your slide. So remember, you're going to be using the pre-cut filter papers, and you want to keep these moist the whole time. So why are we heating? Why are we heating these up? Acid fast have mycolic acid in their cell walls. It's waxy. What happens when you heat wax? It melts. And so then the dye gets trapped in it, right? You ever melt a candle, right, and something gets caught in that wax and you don't get it out before it cools? It's stuck, right? So this helps facilitate sticking that dye into that waxy cell wall. So over a steam bath, and crank those babies up because we need them steaming. Crank them up all the way if they're not already steaming. We need steam, right, to facilitate. 10, yes, all the way up. High or 10. So our acid-fast negative cells stained with the carbofuchsin, that's the first dye we use, right, over the heated plate. If they're acid-fast negative, are they going to get stained with the carbofuchsin? Well, this is a differential stain, just like gram stain. Do gram negatives get stained with the crystal violet? Yeah, but they don't stay stained, right? What do we do to remove the stain? Right, we decolorize. In the case of gram stain, it's 95% ethanol. In the case of the acid-fast stain, it's going to be acid alcohol. And so then again, you've got to do what after you decolorize? You got to counter stain, rinse with water, right? Counter stain, and what's the counter stain we use for the acid-fast Zielen-Neeson method in this lab? Methylene blue. Why do you suppose the acid-fast stain is not widely used as the gram stain? Right. Only acid-fast bacilli, for the most part, are going to stain acid-fast, right? Basically, your mycobacteria. That's not a lot of bacteria, right? That's one genus that doesn't have a ton of members. So most of the time, as you guys are going to see today, you're going to get non-acid-fast. So you're back to square one, basically, right? If it's non-acid-fast. If you got lucky and it's acid-fast, you're like, ooh, I just narrowed it down to mycobacteria or maybe a few others. Right? Where gram stain, what do we usually end up with, right? If I give you an organism, what's the chance it's going to be gram positive? 40% of the time it's gram positive. 60% of the time it's gram negative. The possibilities basically got narrowed in half, right? So amongst the population of bacteria, right, gram stain is going to really help us narrow down our possibilities in half, where acid-fast is too narrow, right? It's such a small group. So our steps, right? Mm-hmm.
Should we be wearing goggles with this heat right next to us? Or this chemical?
Only when we start putting dye over it would you have the potential of it splashing into your eyes. So you don't have to have them on right now. It's not sputtering. It will sputter soon. If you feel more comfortable, put them on. I won't ever say no to that. Okay. So for our acid-fast, right, carbofuchsin, when you decolorize, they stay stained, right? When we counter stain, again, in this case, it's not going to be enough to change its color, right? It's going to stay that acid-fast color. Where our non-acid-fast will get stained, but then they'll get decolorized, and so therefore we have to counter stain them with the methylene blue, or we wouldn't see them. But you guys definitely got to do that. So you see something like this, right? So see how it really clumped together there? But here in this picture you see them by themselves, right? You're just going to see non-acid fast. You should just see blue cells, right? We don't have any acid fast. So an endospore, as we mentioned earlier, is not a form of reproduction. This is important. This is a dormant cell type that only some bacteria can form. And that's why we're using the Bacillus for this, because this is a known spore former. They're protecting their DNA when they do this. A lot of organisms that have this ability are also known as saprophytes. So if you look this up in the back of your book, it said it was a heterotroph that digests dead organic matter. So there are decomposers, right, in the soil. So basically when food is available, they eat it, and when it's not, they turn into an endospore waiting for the next meal, right? But what's a heterotroph? You guys wrote down that definition and you didn't look up the one you didn't know? Shame on you. Heterotrophs are organisms that require carbon in an organic form. So in other words, they eat other organisms that were once living. They can't get carbon dioxide from the air and be able to make their own carbon molecules. They got to eat somebody else. In this case, they're specific in that the somebody else is dead. Saprophytes only eat dead stuff. That would be us, right? You eat anything that's still alive? Not usually, right? It's not like Walking Dead zombies, right? So why do we have to heat this one too, right? So we're going to have to heat the endospore one too. So what's the reason that we heat this one? The reason is that the protein keratin, it packs in between the cell wall layers when it makes the endospore. So it's really hard to stain an endospore without using heat to kind of weaken that protein, that keratin layer, and really get the dye into that endospore. Right? And so this is the same stuff, right? This is the same protein that makes our nails hard, our hair, the outer layer of our skin water resistant. For other animals, it what makes up their hooves and their horns, even the shells of some animals. So again, why do we want to know if it's a spore producer? Well, some of them are really bad guys, right? They're pathogenic. There are 2 genera that contain a lot of spore formers that can cause a lot of problems. So Bacillus anthracis, obviously we're not going to use it, is a spore former, right? That's how they were able to spread that powder. That powder, y'all, that went through the mail was millions upon billions of endospores. They weren't actively metabolizing, they were resting until that person inhaled it. And then they grew into the original cell they once were and started eating the person, literally, and killed them. So Clostridium tetani is another one we worry about. This spore is very common in the environment and soil, and so we could possibly be exposed to it. So what do we do to protect ourselves against the toxin that this one produces? We get our vaccine boosted every 10 years for tetanus. No, this is right. None of these pathogens. Am I going to ask you specific examples about it? This is for your information moving forward in your careers. So Clostridium, right? It's another spore former. Clostridium botulism, another toxin producer, right? Can cause really serious food poisoning. So we definitely want to know, right, if an organism is a spore producer. Clostridium perfringens, right, which can cause gas gangrene. So these are ones that you'll hear, right, if you continue on in the medical field, that you obviously don't want to mess with if you don't have to. Clostridium difficile, right, what people commonly abbreviate C. diff, right, is another one that's a spore former. It's found in the environment, right, and under certain conditions it can cause a really nasty infection, in this case in the intestines. So we already went over location, right? So central, which picture? A, B, or C, or D is centrally located? A. Terminally located? C and D. Subterminal? B, the only one left. And the spore itself can be spherical. Which one looks more spherical? D, definitely, right? And then the rest of them are more elliptical, which is what you should see today, right? Where it's kind of an oblong spore. So each cell has a vegetative cycle where they continue to multiply by binary fission. But if conditions become harsh, it'll copy its DNA and package it in the endospore coat. Unfortunately, a lot of the terms sound like reproduction, right? Mother, spore, mother cell, right? It's not, right? These are the ones that produces the spore. And as I said, it's called an endospore because it's within the cell. But eventually the cell itself will deteriorate and you end up with the spore all by itself. So these are just the steps of the process. I'm not going to test you specifically on any of that. So for this procedure, again, heating to facilitate getting the first dye, the malachite green, into the endospore coat. Decolorization step is just with water, lots of water, right? And then you're going to counterstain with safranin to be able to see the cells, right? Because rinsing with water with the malachite green, it'll rinse out of the cells. So be able to see the cells, we want to stain them with safranin. So yours will probably look something like this, but mostly with more of the greenish blue spores all by themselves. You see how they're not in the cells? But you'll know if it's a spore if it's smaller than the cell. So what color are the spores? Right, this greenish blue color. So why are we using the old cultures? We need 5 days or older because they run out of food, so they form endospores. So we'll actually see and stain endospores today. So if it comes out positive, right, we know it's a spore former. What if you mess up and say do your E. coli today for your spore stain? So you see this rod, you see it's red, but you don't see any spores. Does that mean it's not a spore former? Probably, because that's a week-old culture, right? Because you know that it's a week-old culture. What if I gave you a fresh culture of Bacillus from yesterday and put it on your lab bench? Are you going to see spores? Probably not. Right? Because they're happy, they're all germinated, right? They're all growing. You're not going to see any endospores. So a negative for endospore staining is not a definitive negative, unless you're sure that that culture was grown under harsh conditions such that it produces endospores. For the ones that we work with in this lab, if you incubate them for a week, if they haven't produced endospores by then, they're not going to. That's an important point as it relates to when you get your unknowns. Right? One of the questions is, how old is this culture? That's important when it's related to endospore staining, right? You got to work with old cultures to ensure if there are endospores there, you're able to stain. So why don't we do a negative control for this? What should you at least see on your slide if there are no endospores? You should see the cells, right? The cells themselves act as a negative control. I think that's the last one, right? Nope, one more. So for the Bacillus, if we do some other staining procedure and we see something clear that didn't stain on the inside, do you know that that one's an endospore stainer, an endospore producer? So if we did a gram stain, you have this purple cell and there's this big clear spot on the inside. Is that organism an endospore producer? Maybe. You would have to confirm by doing an endospore stain. Because there are other things inside the cells sometimes that won't stain. Right? It'd be a clue that, hey, maybe I need to do an endospore stain. So that is it. Oh, here's my tips for today. So these are going to be hot, these staining screens, right? So I suggest take one of your trays, use your gloved hands, right? Take the whole tray off. Peel your papers off, put them on a paper towel to transport them to the regular trash so you're not dripping dye all over the place, and then distribute the slides to each person, and each person finish staining their slide, right? Decolorize and counter stain on their staining